Okay, today I'm going to talk about the guidelines for biological drawing. What are some of the things that we need to take note? We need to take note of when we are doing biological drawing. Okay, so first thing first, P. Okay, pencil. So whenever we do any biological drawing, we need to use a pencil. Everything, including the labeling, should be in pencil. Okay, so when you use a pencil, it should be sharp and at least 2B. Okay, at least the pencil must be at least a 2B pencil. Okay, so that it will not be the drawing, the lines that you're drawing will not be too faint and it cannot be seen clearly. Okay, so that is the first P. Then second P that you need to remember is proportion. So whatever we draw, okay, most likely there will be different parts. Okay, there will be different parts in a drawing, or a drawing may have different components. So you need to ensure that the different components are in correct proportion to each other. Okay, so when we talk about proportion, we are talking about the different components. Okay, different components in the drawing, and we must ensure that they have the correct relative, okay, have correct relative proportion to each other. Okay, correct relative proportion to one another. Okay, so let's look at an example. Okay, I'm going to show you what do I mean by proportion. Okay, so let's look at how to draw a human cheek cell. Okay, so on the left side of the screen, you can see a few human cheek cells. Okay, so this is taken under a light microscope. So when you view, okay, when you look into a light microscope and then you see 4 different human cheek cells. Okay, before you start drawing, okay, you need to look at all the cells that you can see. Okay, all the human cheek cells that you can see, and then you decide which one you want to draw. Okay, so for instance, in this case, these 4 cells, okay, I will focus on the one at the center. Okay, then after I select the one cell that I want to focus on, okay, before I start drawing, I need to also look at the other cells, because the drawing that I am drawing, it must shows the main characteristics, the main details that is present in all, if not most, of the cells that I'm looking at. Okay, so that's one thing. Then secondly, you need to read the question. Sometimes the question wants you to draw just one cheek cell. Okay, they will phrase it like draw one human cheek cell or draw a human cheek cell. Okay, sometimes they may need you to draw a few cells, or they may state very clearly, I want you to draw 3 cells, 2 cells, or 5 cells. So you need to read the question very carefully. Okay, so imagine now the question wants you to draw a human cheek cell, that means 1. Okay, so I will focus on one of the human cheek cell. Then I'll look at all the other cells also, so that in my mind, I know what are the main things that I want to draw. Okay, so in this human cheek cell, in this diagram, okay, so this is how I will draw. Okay, so first I will draw the outline of the cell. Okay, some of you will say, hey, the outline that you draw is so different from the one. Okay, if you look at the human cheek cell, okay, it is roughly, I wouldn't say circular, okay, but then I also wouldn't say that it's oval in shape, okay. So it's roughly rounded. So I just need to draw a rounded um, uh, shape, okay. So after I draw the outline, okay, then the next most clear thing that I can see will be this thing at the center, okay. So I will also draw it about near the center, okay. So this is how I will draw a cell. How I will draw a human trick cell. So if you look at my drawing, okay, it is in proportion, okay. So the nucleus, by the way, this is the nucleus. So I need to label the nucleus. Okay, so this is the nucleus. Then this is the cell membrane. So I don't have space on the right side, so I'll label on the left side. Okay, this is the cell membrane. Then I can also label the cytoplasm. Okay, okay. So these are the three things that I can label. Okay, later I will talk more about the labeling. Okay, so now you can see for yourself, when I draw the human trick cell, I need to draw the nucleus in proportion to the whole cell. Okay, so students or anyone, if you draw this drawing, some of you will commit this mistake. Okay, you will draw outline and then you draw your nucleus. Okay, so in this case, the nucleus is too big. Okay, the nucleus is not in proportion to the whole cell, so this is wrong. Okay, so this is something that you need to take note. Okay, so this is the first example. Okay, then the second thing that you need to take note, the second thing that we need to take note is L. Okay, L stands for line and labeling and label. Okay, so whenever I do a drawing or whenever you do a drawing, okay, you need to make sure that the line is solid and continuous. Okay, then when we do labeling, you need to use labeling lines. Okay, the labeling line should be straight horizontal lines. So you need to draw the labeling line using a ruler. Okay, horizontal means from left to right. Okay, then for labeling lines, one thing to take note, do not use arrows. Okay, do not use arrows for labeling. Okay, this is wrong. Do not. Okay, then for labeling line, okay, for labels, okay, my recommendation is label to the left and right side. Left and right side of the drawing. So in other words, we try to draw our drawing at the center of the space provided so that we have enough space on the left and right side to actually do our labeling. Okay, so again, let's look at an example. Okay, so now we are going to see use the example so you can see the onion cells. Okay, we are going to try drawing the onion cells. Okay, so again, you choose one of the cell that you want to draw, that you want to focus on and draw. Okay, so in this case, I will choose, okay, it's really up to you. I can choose, okay, I will choose this cell here. Okay, but before you start drawing, again, you need to look at all the onion cells in the slide or what you can see, all the onion cells that you can see under the light microscope. Okay, to identify the main features. Okay, so in this, in under the light microscope or in this photograph, you can see very clearly, you can see the cell wall. Okay, you can also see the nucleus. Then, of course, the cytoplasm. Okay, the cytoplasm and the large central vacuole, there is no clear boundary between the two. Okay, so but you know in between here, okay, all these things that other than the nucleus most likely it is the cytoplasm. Okay, you can't really tell where is the large central vacuole. Okay, it's not very clear on this photograph, so we are not going to draw it. Okay, so when you draw this onion cell, so imagine you need to do labeling. So just now I say already, if you have a space provided, okay, imagine this box is the space provided. Okay, this is the box provided. So this is the space provided. So when I need to draw an onion cell, okay, I will try to draw at the center. Okay, and at the same time, I will leave enough space. It is a conscious effort. You must leave enough space on the left side and the right side for you to do labeling. Okay, so I need to draw, okay, the onion cell. Okay, it looks roughly rectangular in shape. Okay, so I draw the outline first. So again, you notice that I use one stroke to draw the outline. Okay, because this will ensure that my line is solid and continuous. Okay, I don't draw a small bit and then take out. Okay, so later I will talk a bit more about that. Okay, so after I draw the outline, okay, then I draw the cell wall. So again, when I draw the cell wall, one stroke. Okay, then next, the last thing that I will draw will be the nucleus. Okay, so nucleus, I can, it is near the cell wall. Okay, it's near the edge of the cell wall. So I can choose to draw, okay, somewhere here. Okay. So I will draw the nucleus somewhere around here. So after you have drawn, okay, after you have checked that your drawing is good enough, okay, you a solid continuous line, you have drawn all the main features or the main details that you can see, then you start labeling. Okay, so always label to the left and right side. Okay, try to draw not very long labeling lines. Okay, so for instance, in this case, I will label the cell wall on the right side. Okay, do you see that my labeling line is actually very short? Okay, so I'll label the cell wall first. Then, of course, if I can label the cell wall, I also know that the cell membrane will be pushed against the cell wall. So I can also label the cell membrane. Okay. Then after I'm done with this, my left side of the diagram of the space provided, no space ready. I cannot do any labeling already. So I move to the left side. Okay, so of course I will label the nucleus first. Okay, I don't have a ruler at the moment. So anyway, it's very difficult to use a ruler on the screen. So remember, it must be, the labeling lines must be straight and horizontal. Okay, I try to draw it in a straight and horizontal manner. Okay, and the last thing that I can label will be the cytoplasm. Okay, so this is how you should do your labeling. Okay, make sure that the drawing is at the center, and you can do the labeling on the left side and the right side. Then some of you will ask, how come cannot label on top? How come cannot label below? Okay, if you want to label on top and below, it is also, if you really need the space, then it's all right, go ahead. Then if you are doing that, then can you make sure that it's also, your labeling line is also straight, but now it is vertical. Okay, so for instance, I'll show you an example. So for instance, I can also label the cell wall. Okay, then I also can label the cell membrane. Okay, so I emphasize again, try to label to the left side and right side of your drawing. Okay, if you really need, okay, if you really don't have space to label on the left and right side, then you label on the top and bottom. Okay, and when you label on the top and bottom, then you use your labeling line should be straight vertical lines. Okay, so that is something that for you to take note. Okay, so that is the second thing that you need to take note of. The third thing, okay, the third thing that you need to take note is the S. Okay, it's S. Okay, so when we talk about S, is the size, the drawing, as well as shading or coloring. Okay, so when we make a drawing, okay, we must make sure that it is large enough. So size-wise, preferably at least half the space provided. Half the space provided. Okay. However, some people, some teacher may be more strict. Okay, they, instead of looking at half, some teachers or some people will be emphasizing. They might even say 2/3 of the space provided or 3/4 of the space provided. So in general, just make sure that your drawing is large enough. Okay, why do we have this guideline? Okay, on size, like at least half, at least 2/3, at least 3/4, because we don't want students to draw a very, very small drawing, then we cannot see anything. Okay, so the main point of this size is to make sure that you draw a large drawing. Okay, large enough for us to see the details. Okay, for people to see the details of your drawing clearly, okay, easily. Okay, so it's the main thing is a large drawing. Okay, so this is something for you to, so it's at least half, 2/3 or 3/4. Then shading and coloring is not allowed unless required by the question. Okay, it's not allowed. So do not color your drawing, do not shade your drawing. Okay, so it's not allowed unless, okay, requested by the question or unless specified by the question. Okay, so sometimes the question will tell you, say, to mark out a certain region. Okay, and then if they ask you to do shading, then of course you need to follow. Okay, usually that doesn't happen. Okay, so in general, for your syllabus, shading and coloring is not allowed. Okay, so again, I will use an example. Okay, use an example. So for instance, I have to draw a human cheek cell, and then this is the space provided. The box is the space provided. Okay, so you must make sure you draw a big drawing. Okay, some students they will go and draw the human cheek cell so small. Okay, this is not allowed. Too small. Okay, then some students are okay, another extreme. Okay, when we ask them to draw a large drawing, they draw a very, very large drawing. Okay, so some of them might do this. Okay, I exaggerate a bit. So imagine there is a box drawn for you. Okay, try to keep within the box. Okay, even though we tell you you must draw a big drawing, a large drawing. Okay, some students will draw until too big. Okay, this is also not allowed. Okay, this is also not recommended because your drawing and the line for the box would cut each other. Okay, so that is that will make your drawing very messy. Okay, looks messy. So don't draw too big, don't draw too small. Okay, this is too big. Okay, so the best way is to, okay, the best size. So again, if you're given a box, draw at the center. Okay, then you try to draw as big as you can. Okay, so this is a big drawing. Okay, this is recommended. Okay, this is the recommended one. Okay, so this is how you decide. So if you're given a space, make as big a drawing as possible, but not too big and not too small. Okay, then I also want to talk a bit more about the shading. Okay, so I will use another example. Sometimes they will ask you to draw a leaf. Okay, so imagine you are given a leaf, and okay, this is not very good, so I redraw. So imagine you are given a leaf, and you are given a variegated leaf. Okay, so what is a variegated leaf? Okay, variegated leaf, they have green parts, and they also have non-green parts. So the green parts, they contain chlorophyll. Okay, they contain chloroplast, which contains chlorophyll. That's why they look green. Okay, they contain chlorophyll. So usually in your syllabus, they may ask you to carry out the test for starch in a variegated leaf. Okay, then one of the popular question is they will ask you to do, ask you to do is to test for the presence of starch in a variegated leaf, and then they will ask you to draw. Okay, so then you will need to indicate the regions that is blue-black, that will turn blue-black, the regions that will remain yellowish-brown. So for instance, if this variegated leaf, take it for example, okay, the variegated leaf looks something like this. Okay, so it's not good. Okay, let me redraw it. Okay, so for instance, the outer region is non-green. Then the inner region is okay, green parts of the leaf. Okay, so when you carry out the starch test, the test for starch, test for the presence of starch in a variegated leaf. Okay, if the leaf, okay, or the plant which the leaf is found is plucked from, okay, have all the necessary conditions for photosynthesis to occur. Then if you test for the presence of starch in this variegated leaf, okay, and then you need to draw. Okay, so again, draw a box to signify the space provided. Okay, so you will draw the leaf. Then you will draw the outline. So the outer region will remain yellowish brown. Actually not remain yellowish brown, okay? Wrong description. Okay, it will be stained yellowish brown by the iodine solution. Iodine solution is yellowish brown. Okay, then the one at the center, okay, it will be stained blue-black. okay, because of the presence of starch. Okay, because of the presence of starch in the green areas, in the green parts, and the absence of starch in the non-green parts. Okay, so in this case, you do not do shading. Okay, some students will start to shade. Okay, they will shade, okay, shade the blue-black region. Okay, it is not allowed. Okay, don't need to do the shading. You just draw the outline and then you just label the different regions clearly. Okay, like what I have done. Okay, to the left side and the right side. And again, the labeling lines must be straight horizontal lines. Okay, so maybe just do a summary. So whenever we do biological drawing, okay, so whenever you do biological drawing, remember PLS, okay, P, L, and S. Okay, P means draw with pencil, correct proportion. Then L is for the line. Okay, remember the line must be solid, continuous line. Okay, broken lines are so just to add a bit more on the solid continuous line. Okay, things like, okay, for instance, in this case, so if you don't use a one stroke to do your drawing, okay, if you do this type of things like what I'm doing now, okay, I draw already. Okay, I draw part of it and then I take my hand up, then I start drawing, then I draw again. So if I don't use one stroke, okay, there is this possibility of having broken lines or overlapping lines. All this will make your drawing untidy. Okay, so you need to avoid broken lines. You need to avoid overlapping lines. Okay, that is also why it's solid. Why my advice is one stroke. Okay, try when you do the drawing, use one stroke to finish the drawing. Okay, every time for each part. Okay, so solid continuous lines, so avoid overlapping lines, avoid broken lines. Then labeling, the second L is label. Okay, remember to the left side and right side. Left and right side of the drawing. Okay, then for your label, make sure it is a straight horizontal line. Okay, do not use arrows. Okay, label, no arrows. Okay, no arrows. Okay, I say no arrows and I use arrow. Okay, so no arrows. Okay, do not use arrows. Then for the S, okay, the size, at least half the space provided. Okay, in general, at least half should be good enough. Okay, but just now I already mentioned already. Size basically just means that make sure your drawing is large enough and so that the details are clear. Okay, as long as it's large enough and whatever details can be seen clearly, it is good enough. Okay, so at least half is just to guide you. Then the other S will be the shading. No shading and coloring unless the question asks you for it. Okay, no shading is allowed. So that's basically about it. So whenever you do biological drawing, just remember PLS. Okay, PLS. Hopefully this will help you to remember, okay, the important things to take note when you are doing biological drawing. Okay.